Okay, buenas tardes con todos. Hey Daniel, do you hear me? Do you hear me?
I can hear you, yes.
Okay, yeah. Just give me like 2 minutes for talking in Spanish and then I'm gonna switch to English, okay? Okay. So, buenas tardes con todos. Bienvenidos a la tercera sesión de nuestro primer día en este primer Simposio Internacional de Biología Sintética que estamos organizando en la comunidad de Biología Sintética de la Universidad Nacional de San Marcos. Mi nombre es Mijailo Espinosa Fernández, voy a ser el moderador. Y bueno, nuestro objetivo hoy y bueno, en todas estas sesiones va a ser conectar con líderes globales, mundiales, que están revolucionando el campo de la biología sintética. Y bueno, para esta sesión nos acompaña un investigador excepcional desde Alemania, que de hecho tuve la oportunidad de haberlo conocido en China. De hecho, es una bendición de haberlo conocido, ya van a saber por qué. El cual le agradecemos enormemente por esta oportunidad, ya que allá es de noche en este momento. Y bueno, antes de seguir también quería comentarles que cada vez que dejen una pregunta en el chat, o bueno, también lo hagan al momento de pasar a ese tópico, que, bueno, se va a contar para que participen en un sorteo del certificado. Esto auspiciado por Biolab Scientific. Y bueno, a partir de ahora voy a cambiar a inglés, así que, bueno, good afternoon, everyone, and good evening to our special guest. It's my absolute privilege to introduce Dr. Daniel Schindler. Dr. Schindler completed his PhD at Micro in Germany and advanced his postdoctoral research in Gishi's Kai's group. After leading the Max Genesis Biofoundry at the Max Planck Institute, he recently became a full professor at the Center for Molecular Biology of Heidelberg University. His lab uses synthetic biology and synthetic genomics in a fascinating build-to-understand approach. On a personal note, like I said in Spanish, I had the immense honor of being his student at the Cold Spring Harbor Asia program, and I can assure you his vision is truly inspiring. Today, he will present synthetic biology and synthetic genomics to engineer and understand the microbes of tomorrow. Daniel, the audience is completely yours. Again, thank you so much for being here, and well, let's start.
Okay, thank you for inviting me, Michael. It was really nice to see you in China and now again online. Let me share the screen that everything works out. But we should be able to start quickly. Okay, I need to arrange a bit here the windows that I see a bit, but not too much. So please give me a second for that. If you later have questions, most likely Michael has already said that you can also ask them in Spanish, and Michael will most likely translate, or someone else. So feel free to also just do that. And Michael, most likely you will be able to give a bit of a view what's going on in the chat, because I maybe don't see everything, right?
Yeah, it's okay. It's okay.
Good. And for all of you, it's already 9 p.m. here, so please bear with me when I'm already a bit tired. But yeah, anyways, so let's start with the topic of today. I'm going to talk about synthetic biology and synthetic genomics to engineer and understand the microbes of tomorrow. And it's more or less like the vision we have in our team and where we and the community would like to drive over the next years. So the goal would be to design synthetic microbes, build their genomes, and then actually do that. But before we come to that point, I would like to show you where I'm actually are at the moment. So this is kind of the sunset at the castle of Heidelberg, where here's the old ruin, and here's a really nice terrace where you can sit in the summer, enjoy the afternoon, and look at the campus where the pointer is roughly the research campus, and along here that line is like the old town, which is a really beautiful city, and the houses there are partially from 1500. So it's a really old town, and it's also the oldest university in Germany. So whenever you have the chance to visit, I can highly recommend that. And now a bit more about the research background where we are. So we at the Center for Molecular Biology of Heidelberg University. That one was created in the 1980s, and it had like a new vision to kind of make researchers again a bit more independent, that the research groups are not gigantically big, but that they are really effective and that they can focus on research and that they have a bit more of independence how they can manage things. So there's kind of a dedicated research and training program, and it's always kind of in the molecular biology research and coupled to biomedical research, because in Heidelberg, there's also the German Cancer Research Center, where there's, I don't know exactly how many researchers, but it's like one of the main topics here. And our centers has always around 250 to 300 employees in 15 different research groups. And currently that's the composition. Very unfortunately, we had an initiative to found Germany's first Center for Synthetic Genomics. It's called Center SynGen, and it is in total at 3 locations, one of them in Heidelberg University, one is in Karlsruhe at the Karlsruhe Institute for Technology, and one at the University of Mainz. Only in Heidelberg for the first year, for the first 5 years, they invested 5 million euros to kickstart this initiative. And we have a biofoundry established, which is now led by Konrad Herbst. And we had a recruitment of 2 full professors and 1 junior group, and one of them was me within this topic. So that being said, I just moved last summer to Heidelberg, and this is still the team of mine in Marburg, where we're at the Max Planck Institute. So the picture is a bit older, but we are a very international team, spanning more or less the whole world, and it's constantly growing. And we are very excited to always host interested people whenever possible. And our approach is basically that we design DNA on a computer, then we build it in vitro, and then we bring it into cells to ultimately check their functionality. That can be towards biotechnological applications, or it can be mechanistic understanding, for example, building whole synthetic chromosomes. And we do that in the synthetic biology design, build, test, learn cycle, which I don't need to introduce to this audience, I guess. There's different aspects, and it's a very interdisciplinary topic. For my talk today, I would like to give some definitions, which is always good. So when I talk today about synthetic biology, then I mean about the part of biology that you see your systems from the perspective of an engineer, that you use standardized parts and chassis to build and understand biology, or to manufacture something towards biotechnology. And synthetic genomics is the global restructuring of large chromosomal segments, whole chromosomes, and whole genomes to understand the basic principles of chromosome biology. And here the point is, I would say that's at the moment still very on the basic research end, but maybe in 5-10 years that will also kick off in real applications. Most likely you have heard about companies like Ginkgo, which really try to put that into high automation elements. And that's also what are the core elements in synthetic biology and synthetic genomics: DNA synthesis, standardized DNA assembly, and automation compatible processes, because you scale things up. And how that works is what I'm going to introduce now. So the DNA synthesis is still expensive, and a lot of people now use standardized assemblies that you can create with type 2S restriction enzymes, part libraries, which you then can combine in a certain way. And they work that those enzymes have a recognition site, which is indicated here. Then they recognize this directed one, but they cut slightly outside. And then you end up with 2 fragments, one fragment which has the recognition site, and one which has it not. But they have these overhangs, and if you design that properly, you can do an assembly system out of it. Often it's called Golden Gate cloning. There's also other terms, but Golden Gate is kind of the standard. And here is now in green depicted the enzyme with the recognition site, and in the color part, you have the different overhangs, because it can be any sequence here. It does not need to be CGG, that can also be ATG or something else. So here now, I use colors: orange, greenish, bluish, and purple. And you see directly that this green part of element 1 fits to the green part of element 2, and the blue one to element 3. And if you have now an acceptor vector where those sequences are just inverted, they cut to the outside. And that means when you have also now the restriction or the selection markers, which are here indicated by a blue color and here by brown, you can put all of them into one reaction. You add the ligase, you add the restriction enzyme, and over time, you enrich your assembled plasmid where you select for the brown marker, and then you have them assembled into 1, 2, and 3. And now the enzyme cannot cut this construct anymore, and you come to the point where you have a piece of DNA which you wanted to have. This is now very abstract with numbers, but all of us know that if you replace the number 1 for promoter, number 2 for coding sequence, and number 3 for terminator, then you come to a transcription unit. So what I've showed before basically is that you do your design on a computer, you generate your level 0 parts or basic parts, which is then the part number 1, 2, 3, or however complex your system is. And those you can then assemble into transcription units. And if you use the right enzymes, you can even release those individual transcription units and can combine multiple of them. And that's a technology we use in the lab to build really large pathways to be able to go to chromosome-sized elements. And now this might be, for some of you, sounding quite abstract, but what I want to show you is that we use then automation, because when you handle a lot of different parts, you go away from your single tubes because it's getting more and more complicated. Most of you have pipetted a 96 well plate. I doubt many of you have done a 384 well plate, and none of you has done a 1536 well plate. And in this area here, you can still do it manually. Here it's impossible, but already here it's hard to find the right candidate or the right reaction when you just go with a pipette to your well M3, or M3 might be cool, but how about C17? So you need to count very carefully, and you have the chance that you miss it up completely. So we use automation, and also when you use automation, you need to be sustainable. So what we use a lot is kind of pinning robots, where we have here a transfer plate with 384 pins, which transfers from one of those plates a tiny droplet to something else. And that's something you really need to consider, because otherwise you run into like high extreme costs. And what we have done over the last years at the Max Planck Institute, and already before in my postdoc time, we have established workflows to build DNA. So we start with an acoustic dispenser. That machine allows you to downscale your reaction to 1 microliter total volume. How that works exactly, I will show in the next slide. So this does our reaction. We put them into the PCR cycler, and our PCR cyclers do then 2 times 384 reactions in parallel. Then we do the transformation into E. coli, and we can select our candidates with a colony picking robot. Those get transferred in a standardized format, and then we use a screening robot to transfer them from A to B. And then at the end, we do nanopore sequencing of the candidates which interest us, but most of the time we sequence by now everything, because we also learn from the things which do not work. I come now back to this machine here. Most of you have never heard about it. I would recommend to use this video you can find on YouTube, which is a very nice illustration of how the system works. How it actually works is that you have here a tiny door which can open, and then you slide in your initial plate. I hope you can see me on the screen now, where your source parts are in. That's basically this element here with the liquids, and then inverted on top, you have the destination plate. And below the source plate, you have a transducer. To say it simple, it's a very sophisticated music box which ejects sound waves, and those sound waves get reflected by physical laws. So you can calculate where the liquid height is, and you would do that for every well, and then you give the machine a list of coordinates, and then it uses a focused sound beam to transfer tiny droplets which are flying up and sticking to the top plate. For those of you who are really interested, I really recommend to watch that video, because this illustration from the company is pretty good. So we do our reactions now with this machine, because we can do the transfer of DNA, of enzymes, of buffers in very small volumes, so that in end, our reaction is 1 microliter total volume. Then we put that into the PCR cycler, where we run our reactions through. Those reactions we then transform into E. coli. And already when you think now the transformation of a 96 well plate is quite intense, yes, it is. You need to go away from a single transformation plate. So what we usually do is we take a multi-channel pipette, which has 12 channels, and we put a tiny droplet on the top of the plate, and then we tilt the plate so that the drops run down along the plate. And that means each of those lanes is one transformation. And in this way, we manage to separate already 12 transformation on a single agar plate, which reduces the number of plates by the factor of 12. But if we go really high throughput, even that is not sufficient, because a 384 well plate would be still a bunch of 12 lane plates, plus the risk of mixing it up. So we can move even further, that we use this pinning robot here and do this transformation basically in 96 well plates. So what you see here now most likely is a confusing image, but if I put now a grid on top of it, you see that there's tiny boxes, and each of those boxes is a transfer from one multi-titer plate well to this agar plate. And what we do is we set a droplet on the left, and then we do 6 more droplets to the right. And then we go to a revisit of the original source plate. We go drop down and then do again the 6 transfers, and then the same and the same. So it's 7 times 7 spots. And with this, we arrange that we have a dilution series where we have a high concentration on the left and a lower concentration on the right. And in most of the transformations, you can also see that we really have the colonies there. So this allows us to go in terms of those transformations into really a high throughput format, which is then also easy to handle because those plates, we can feed them into our colony picking robot. And this robot recognizes the colonies. This is now an example plate where it takes a colony, it transfers it to the destination plate where it goes into a standardized format. And then afterwards, a tiny bit of a plastic filament is chopped off. So it's like a fishing line which goes through the machine, which is sterilized. And then whenever you transfer a colony, 2 millimeters or 3 millimeters of this filament is chopped off, and then it transfers to the next one. And once you do that experiment, you end up with a plate where you then have, in this case, 384 different yeast colonies, and you can work on again with these plates on your screening robot, as I've shown before. And the screening robot then actually looks like this. So you see on the right here, an arm which takes one of those pipettes. Inside, there are arms which take away the lid of the plate. It dips into the first plate, and then it transfers it here into a liquid culture plate. And then afterwards, it drops the plate here, and then the video restarts again. And with this, we can go very sustainable into really high throughput formats because those pipettes, they can be recycled, and each of them is basically 4 pipette box or pipette tip boxes. So because we can use those pipettes over and over again, we save massively on the plastic waste and also in terms of the funding we need. And to show you now an example of this, we have done one experiment where we created more than 2,500 different E. coli candidates, each of them producing potentially one new natural product. And they are grown here in 384 well plates, where we have collection 1 here, collection 2 here, collection 3 here, 4, 5, 6, and 7. And then we grow them under 8 different conditions in total. So we have the control condition, we have a condition where we mildly induce it, we have it medium induced, we have it stronger induced, we have some additional compounds in, and so on and so forth. And then we take those plates, we put them into a plate reader, into a stack, and then it measures all of the individual wells, and it tells us basically at what time point the production of what we have in the cells kills the producing host. So as soon as we know our product kills E. coli, we know that that's a potential new antibiotic. And at the bottom here, there's the same experiment, but this time with the solid agar plates as I've shown before. And to give you an idea how that looks, so we have now the 384 well format where we have this pinning in tiny patches again. So this time is 9 spots, so basically 3 to the right and 3 down, plus the lines. And the red box here is the control. This is how it looks under the standard conditions when there's no effect. And when we put it to the expression conditions or something's in the media, then we all of a sudden see that certain candidates are dying and that they are not performing that well anymore. And here we can then use nanopore sequencing to sequence all colonies based on what kind of plasmid they have inside or whatever genetic modification we have done. And we would sequence the one which is of interest because they have lost the features, or even the ones which are still alive here, because then we can understand if there was an error in the construct or if there's something else that the cells are resistant to it. So we can produce a lot of data, and also now in the time of machine learning and big data handling, you can really increase your knowledge gain here. So, and this pipeline basically runs in the back of all our projects. So we usually don't highlight that anymore. It's only for people to really visualize what's behind that, because in the end, when we talk about the research, you don't see so much of this anymore. And to give you an idea what we are doing in the lab, we have 3 major project lines. So on the one hand, we are interested on small regulatory RNAs. They are in bacteria, and you can engineer them to make specific functions. It's basically a regulatory tool which acts after the level of transcription because you prevent the production of proteins. But I'm not going to talk about this part today. Then we are interested in non-model microbes. So there's a lot of microbes in our collections everywhere in the world, but most of us just works with Bacillus, E. coli, maybe Caulobacter, and Saccharomyces cerevisiae. But there's so much more diversity, which is also really relevant. And there's some example I will show in the next slides. And then I will go in the end towards the building a synthetic chromosome. So I will only give a snapshot here because this topic on its own would be at least like an hour of talking. So we start now with this element, and that has kind of a background. It's about Vibrio species. And as you can see here in this figure, Vibrio species usually have 2 chromosomes in each cell. And we created a strain which has them all fused. And why we work with Vibrio is because Vibrio is a very interesting species. The majority of them are pathogens. So there's Vibrio cholerae, for example. There are some animal pathogens and so on and so forth. But there's also some which are non-pathogenic. So they're classified as biosafety level 1. They grow under the same conditions as E. coli, shown here on the right. And this organism basically only needs salt on top of the normal growth. But it has a really interesting feature because it's the fastest grown microbe today. So you can see that this microbe really grows rapidly. So when E. coli did a couple of cell divisions, this one has kind of filled all the chamber already. And under optimal conditions, it's reported that this organism has a doubling time of below 10 minutes. In our plate reader assays, we come to the point that we get it towards 12 to 13 minutes, but it's not the optimal conditions. But still, it's much faster than E. coli. And for that reason, we thought at a certain point it would be really cool to take this organism, to fuse the 2 chromosomes, and then use the second chromosome because it has a separate origin of replication. So only this element, again, to introduce synthetic chromosomes into this organism for the expression, for example, of biosynthetic pathways. And to do that, actually, we took a sequence area from here and we integrated that with our CRISPR-Cas systems around this origin of replication. And then we forced to make a double strand break here at the origin of replication so that this kind of sequences recombines here and creates a final strain which has only one origin of replication and only one diff site, which is important to separate the chromosomes from each other. And you can see here the fusion sites. At the same time, this origin is lost and this diff site is lost as well. And you can use this to build up a new chromosome within that cell. And we then use different technologies to actually validate our strain. So this is now pulse field gel electrophoresis, where you do a DNA extraction very gentle in agar plug, and then you run the DNA very slowly in zigzag through the gel. So it takes most of the time many, many hours, most often days, that this chromosome separate. And you can see here is this blob of the chromosome 1, which is on roughly 3 megabases. You have here chromosome 2, which is roughly 2 megabases. This is the parental strain. And in the strain where we fuse the chromosome, we see that here at the expected height, we have a band appearing, which is the addition of those 2. So we can show in this gel that this strain was created because also the other bands on that height here are disappearing. And we also proved that with nanopore sequencing, that we sequenced the whole genomes and then we aligned the reads to this area and to this area. And we see that we have consecutive reads in this area, which shows that it's fused at that point. We also took the reads of this strain and mapped them to the original chromosomes. And then we see that we have the deletion of the diff site here and that it's not a continuous read anymore. And we also see the loss of this origin region. And one of the interesting parts here is already when we were preparing the DNA to validate the strains for sequencing, we realized that the strain was not so much growing slower. And we confirmed that later with different assays that the parental strain and the strain with the fused chromosome, they have the same doubling time in our plate reader assay, which is only a difference of 3 to 5%-ish. But they're growing more or less at the same pace. And you can see E. coli here as an outlier, how much slower it is. And if you put that into like a normal growth curve, you also see that the growth curve is more or less the same. They reach the same density. And what I always like to point out is that after 2 hours, you're in early exponential phase. And after 4 hours, you're done with your experiment because you reach stationary phase. So you can run 3 to 4 experiments a day with this organism if you really want to do that. We also tested different stress conditions to show that the DNA replication is not having any problems because of the fusion. And we also checked the cell morphology, and we basically see no differences when we look at single cell level. If we take a lot of those cells, we see that the synthetic cells are slightly wider and slightly longer, but that's really a minor thing. And if you look at individual pictures, you would not really see that. Okay, then we come to an experiment which is kind of very interesting because we wanted to prove that there's also only one origin of replication left. And we have done replication pattern analysis where we have on the x-axis now the individual chromosomes on the genome part. On the y-axis, we have the relative copy number abundance of each of the alleles we have sequenced. So we sequenced one sample where we had a stationary phase culture and one with an exponentially growing one. And then when you plot that and normalize it, you get like this standard plots that where you have the peak, you have the origin of replication of this organism. And as expected in the wild type, we have the origin of replication for the main chromosome here and for the second chromosome here. The red line is kind of a regulatory sequence. Once this is replicated, the initiation of this chromosome takes place. And we have done the same in our single chromosome strain with the fused chromosome. And we see now that we have the same pattern, that there is only one origin of replication, which means that DNA replication is not the problem to generate the respective speed of the growth. So now I come back to this sequence very shortly because that's important for the synthetic chromosome part. Because when you have this sequence, this element here is initiated and you have this copy number of the chromosome. So now this sequence is highly conserved in Vibrio species. If you delete it at this part here and move it up, then you also move up the copy number of this part. And if you move it down, then you move also the copy number down. So what we are now using in our synthetic strain, where this part is not relevant anymore, we bring in an ORI2-based synthetic chromosome. We can build in any part we want to do, and we can now variate the abundance here because we think that this is kind of a really cool future chassis or platform for expression of interesting products, because this replicon will be maintained in a cell which is used to this replicon. And if we use the positioning aspect of the CRTS, we can control the copy numbers. So we can construct with our automation pipeline a library of those constructs. And at the same time, we can use a library of those strains to very rapidly identify the best expression conditions. And this is an ongoing project, which is not published yet, but we will present some stuff as well on a conference next week. Okay, so with this, I come to the last topic. So the synthetic chromosomes and genomes. And I was fortunate to have been part of the synthetic yeast genome project in the lab of Yisi Kai when I was doing my postdoc, and I constructed one of the synthetic chromosomes. And this is an international effort where many, many labs are involved. And they chose at a certain point Saccharomyces cerevisiae as the model chassis. And the reason for this is because Saccharomyces cerevisiae is the eukaryotic model system. It's very well known because the genome is sequenced since 1996. It has 16 chromosomes. It has mating types, but they can be kept as individual haploids, so you can also do classic genetics. And the major advantage is it has a very efficient homologous recombination machinery. That means yeast usually needs only 30 to 50 nucleotides and can repair a double-strand break or can integrate something into the genome, which gives you a lot of benefits. And the synthetic genome project made now the assumption that we can take this genome, we can recode that based on rational design and design a sequence on the computer, synthesize that, build that, and replace step-by-step the individual chromosomes. And the design has a lot of concepts. So there was integration of PCR tags. That's basically recoded sequences to differentiate the synthetic sequence from the wild type sequence. There was a recoding of the TAG stop codons to be able to, at a certain point, add non-canonical amino acids to this synthetic yeast. Then there was biological questions. Can we remove all introns? Are the repetitive elements important? And obviously, you want to remove the transposons to have a really stable strain. And one additional gimmick was kind of introduced. It's called Scramble, which is an accelerated evolution system because downstream of nearly every non-essential gene, a Cre recombinase site was integrated and allows, after the activation of Cre, that the genome rearranges in a massive amount. And the removal of all tRNAs and placing them onto a neochromosome was one of the central topics because tRNAs are known hotspots of instability, and the idea was to bundle all instability in a single spot to prevent the genomes from being unstable. And this project has been finalized individually by early last year. The 16 individual chromosomes were synthesized each in one strain. So there are 16 strains, one with synthetic chromosome 1, one with synthetic chromosome 2, one with synthetic chromosome 3, and so on and so forth. And afterwards, they can be combined by mating procedures. And by now, there's more than 8 synthetic chromosomes within a single cell. And to construct the final completely synthetic genome, that's still an ongoing endeavor, which I would assume will be finished in the next couple of years. Okay, but first of all, how do you construct such a synthetic chromosome? That's, let's say, relatively simple. You have your wild type chromosome shown on top here, and then we have developed a technology which is called swap-in, and each swap-in round is one round of transformation. And within each round, you transform a piece of DNA, which has a marker, here abstracted in orange, and a homologous region to replace, in this case, this area. And then you select for the synthetic chromosome, which has this element here replaced. In the next round, you take a new fragment, which has a different selection marker. This one is now indicated in green. It has homologous sequences before the other marker. So this one would be eliminated, and this one would be the one you select for. So you have a counter selection back and forth, and you do multiple rounds until you come to the end and have a fully synthetic chromosome. And that was done in various ways to achieve the 16 different synthetic chromosomes. But there you have a template, and there is like one kind of approach where you have a known sequence. So this is kind of different from the project I've done in my postdoc. So I was doing this tRNA neochromosome project. Why do you think about that? tRNAs are really interesting because, you know, you have 64 codons in principle, but you have 275 tRNA genes, which encode for 42 tRNA isoacceptor families. So this 42 different tRNA types recognizes all the different codons, basically. And that means you have a certain redundance. Some are single copy ones, others have more than 10 copies. And interestingly, they are transcribed by RNase polymerase III. So in eukaryotes, you have 3 RNA polymerases, and RNA polymerase III only transcribes tRNA and some other small non-coding RNAs. And they have an internal promoter in the tRNA gene. So the complex binds basically here, and the terminator is a poly-T stretch. So the upstream region here has a regulatory effect, but it's not the promoter you know from mRNAs. What's also really important, tRNAs are 15% of total RNA in the cell, 80% is ribosomes, and tRNAs are much smaller than ribosomal RNAs. That means you have, with tRNAs, most likely one of the most abundant molecules in the cell. All mRNAs, which produce all the proteins, is just 5%. And now tRNAs are scattered all over through the genome, so there's no real patterning behind it. And in yeast, there's also a very particular thing that upstream of tRNA genes, you have quite often transposon insertions because there's an interaction between the RNase polymerase III and the integrase of the transposon, which is a nice and neat biological aspect. So the idea was now to take all these tRNA genes and put them onto a new chromosome to kind of get this instability of transposon insertion away from the individual synthetic chromosomes. And when you do something like this, to design a new chromosome, which has no natural template, you start with a blank page because you really don't know where the starting points are, and you start to look into literature and into your basic molecular biology education, and you realize quickly, well, I need a centromere to be able to segregate it. At the beginning, when it's not essential, I will most likely also need a selection marker, and so on and so forth. And based on this blank page, you come up with a rational design in terms of synthetic biology, and you end up with a very complicated figure, which comes now. But don't worry, we will go there very briefly through. At the x-axis at the bottom, you see the total length. So the chromosome length in the end is 190 kilobases roughly. This is the full design. So there's one of those tRNA neochromosomes, and this consists out of 275 individual tRNA cassettes, which are roughly 700 nucleotides long. They have the individual tRNA gene. They have a 40 nucleotide downstream region. They have a recombination site, which is called ROX, which is similar to LOXP system, but is recognized by a different recombinase. And you have 500 nucleotides of upstream region, which comes from a different organism to prevent homologous recombination with the genome. And then those tRNA arrays or tRNA cassettes are sorted into 16 tRNA arrays, one tRNA array for each of the chromosomes. So this is now chromosome number 1, because it's the smallest chromosome and only has 4 tRNAs. And those individual chromosomes are then ordered into mega arrays, which have then in between always roughly the same spacing as you can see here at the higher level order. And they always have an origin of replication on one side and a synthetic termination site on the other side. And all tRNA transcription and the DNA replication is aligned into one direction so that there's no heads-on collision anymore. And these sequences are then placed into this higher order. And to come to this termination sites, that's a very interesting aspect because there's a system in yeast, which is the FOB1 binding site and the FOB1 protein. They basically prevent that DNA replication can pass through it in one direction, but into the other direction it passes through. So we placed these sequences basically here, but made a double trap out of it so that we have two sites which are kind of letting the DNA replication passing in one direction, blocking it at the second site, and the same from the other direction to then prevent instability in this area. And this chromosome was built, and it was always called the party chromosome because we have put a lot of instability into a single construct. And that was a real journey, which was the major work of one PhD student, Roy Walker, and my postdoc. So we have spent together most likely more than 10 years to finalize this project. Okay, so now how do you build something where you have no template? It cannot be by stepwise replacement, but what we did was basically we ligated the first element into a plasmid. So we have the first array, and then we did the same marker swapping extension here that instead of replacing something, it was something added. So you can continuously see how this element grows. And then at the end, after 15 rounds, you end up with this full element, which has quite a few challenges because also this instability causes cloning issues, as you can imagine. We can now also linearize step constructs because chromosomes in yeast are linear. And for that, we use a system where we have a URA marker. And in this URA marker, there is a recognition site of IC1, which is mega endonuclease. And when you cut this element, the telomeric seed sequences are released, and the telomerase recognizes it and extends that to proper telomeres. And at this stage, when you integrate the URA marker at any position in the genome or in a circular construct, you can select for URA-free. You have basically this construct, and as soon as you activate the expression of IC1, the cut will happen here. You can have no URA-free resistance anymore, but cells become resistant to a chemical compound. So you can directly select for linear chromosomes. And that's what we then have done in this construct. We have selected multiple sites. I don't go too much into details, but what we can see in the end is that here in the controls, you don't see this synthetic chromosome. And once you cut it and linearize it, you can see it in that shell. So we can build a lot of different conformations and we can characterize them. And then we also see in terms of the growth that a wild type strain grows normal. When the chromosome is present, there is a growth defect. The growth defect is most likely because we have the overexpression of 275 highly expressed genes, because when you lose this construct, then the cells become viable again. And then you see the same in the plate reader assay where the wild type control and the lost strain, they grow fine. And the strains with the synthetic chromosomes, they have like growth defects. There's an additional one here, which had a mutation we found later out with genome sequencing. So this is kind of an artifact of the analysis. And we also checked different compounds, in total more than 20 different conditions. And we only found 2 conditions where the growth defect got more severe in the strain. And one is in the presence of azauracil, which has an effect of transcription, and hydroxyurea, which is a compound which is kind of involved in DNA replication, or kind of limits DNA replication. So there's a lot of biological insights we have gained. We have made a really big story out of it. I will not go into the details anymore because that would be really massively over time. So we have done Hi-C analysis. We have done neochromosome stability assays, nuclear localization studies, nucleosome positioning, phenotyping in various ways, proteome analysis, replication pattern analysis, RNA-seq, tRNA-seq. But in general, the story is basically that we can design a chromosome from scratch. We can build it, we can shuffle it around between different yeast strains, and then we can learn a lot. And this is, as you can see, we have only made like the first round to come to the learn process. And we are currently also in the stage that we slowly start to write up the follow-up story, where we have this learning aspect and put that into new designs, a new build, new test, new learn. So we have really used now the synthetic biology concept to bring that to whole chromosome level and to bring that really down the road. So I really hope that this story is also finalizing towards the end of the summer. So maybe in next year when that meeting happens again, I'm happy to talk about that. Okay, with this, I come to the final slide. A lot of acknowledgements. Thank you, Michael, for inviting me. I'm really happy to talk here. Thanks to the team, thanks to the funding bodies, thanks to all the international collaboration partners. Obviously, I have to highlight all the people working in the Synthetic Yeast Genome Project. There's Roy Walker, who did like the hard work together with me to build this tRNA neochromosome, and our supervisor, Patrick Kai, who got all the money to make this work really possible. And it's a really amazing community, and I only can recommend to do that. And with this, I think I'm more or less perfectly on time for closing, and I'm happy to answer your questions if there are any. Thank you.
Thank you, Daniel. Well, it was an amazing presentation. It's fascinating to see the scope of what you were doing in your group, right? Well, so we have actually some questions in the chat. So the first one is from Maria Augusta. She said, "Hi, Daniel, excellent presentation. I like it a lot, and I have a couple of questions for you. In the transformation step during the scale up through DNA, do you mix all the DNA and the competence E. coli in one step?"
So usually we do the reaction in the PCR plate, and then we add competent cells with multi-channel pipettes on top. We have that all on ice, and then the transformation, in this case, we usually do in a PCR cycler. That works pretty well. We have optimized our processes for that. We use a specific homemade type of cells, so that works pretty well.
Yeah, thank you. I mean, actually, she has another question. It says, to construct the synthetic chromosome through recombination, what technique do you use to confirm the recombination occurred correctly?
Okay, so this is a multi-step procedure. So the first confirmation is based on the growth of the selection markers, because we are always swapping back and forth the markers. So the first go is select for the new integration of the new marker. Then we take a couple of colonies, check them if they are growing on the other marker. The next step is then doing a colony PCR to see if the integration flanking sites are giving a const or an amplicon. And then quite often we do whole genome sequencing afterwards. That depends then on the scale at which level we are.
Yeah, perfect. Give me a minute.
Maybe I stop sharing. If I need to go back to the slides, I can do that anytime later, but then I see more than the small black screen.
Ok, perfecto. Okay. Chicos, pueden dejar sus preguntas en el chat en español o en inglés. Bueno, preferiblemente en inglés, pero si lo pueden hacer en español está bien, igual. Y también recuerden que toda pregunta que se realice va a ser contado para el sorteo de certificados. Y también si quieren hablar, pueden simplemente levantar la mano y se le va a prender el audio. If anyone wants to talk, just raise your hand. Okay. Yeah, go ahead.
Thank you, Dr. Daniel, for the presentation. I just had one small question regarding the SC2, the synthetic yeast project. Specifically, I understand that all the tRNAs were removed. So what does happen to the normal gene expression specifically? And to the cellular fitness, is it maintained or is it heavily affected?
So regarding the removal of tRNA genes in the individual chromosomes, that works to a certain extent. You can see that there are certain changes. There are some of the studies which showed afterwards, or maybe let's do it this way. So the synthetic chromosomes individually were designed without having a tRNA integrated anymore. Some of them have a single copy tRNA, which is essential. So you need to supply that in trans somewhere integrated into the genome or via plasmid. Otherwise, the cells would be dead. Then in some of the studies, for example, in synthetic chromosome 2, people have shown that when you have the strain without the tRNAs, it grows fine, but the expression levels change to a certain extent. And if you add a plasmid containing this synthetic tRNA chromosome 2 array, then it normalizes again. So you see aspects because also these highly transcribed genes have effects to the downstream genes. So you change a bit of regulation. But most of the time, it's not looked too much into the details. If that's really interesting for you, most likely there's a lot of data where you can dig in because all this data is publicly available.
Yeah, thank you, Daniel. So there's another question here in chat. It says, Roberto, how could engineer microbes contribute to solving environmental challenges such as pollution or climate change?
I mean, to design that, the most powerful tool we still have in the lab in every way is evolution, right? So if I want to design a microbe which is more resistant to something, I get a tiny bioreactor which has like continuous cultivation, or I get like even flasks and I select for microbes which have an initial growth under this condition or do whatever I want. There's nothing more powerful than evolution. We can design a lot of things. We can have cool ideas. We can transfer a pathway from one organism to the other. There was recently a Nature paper using this rapidly growing microbe I presented in that talk that they put in 3 to 5, I'm not sure, I think 3 or 5 bioremediation pathways into this organism.
Oh.
And then that was like doing a job, which is like actually really cool. That was actually from Yunbiao Dai, Michael, the leader of the Cold Spring Harbor Lab.
Yeah.
Course.
You're talking about Vibrio.
Vibrio, yes.
Right?
Yes.
Okay, cool.
So, and that works with all microbes the best because they're growing so fast that laboratory evolution is something which does amazing things if you set your assays right.
Oh, that's awesome. Okay, perfect. So we have another question here from Arbulu Castillo. He or she says, "Hello, Daniel. Excellent review of the work. My question is, how was managed the tRNA neochromosome stability?"
So the stability is, I would say, relatively normal and behaves like the plasmids in yeast. So you have a loss rate of 20% if you don't have a selection marker. If it gets essential, obviously, then you don't lose it anymore. There are certain aspects which might indicate that they lose it faster because we have a growth defect. And the problem is as soon as you have a growth defect and you select for something which is growing better when it loses it, then you get like a kind of biased into your analysis. So it's relatively tough to analyze. So I would say generally stability is like one of the PS plasmids or the yeast plasmids in general. That's in terms of maintenance, but also because there's a lot of similar sequences, you can have an intrinsic instability that you have DNA recombination within the DNA fragment and certain segments can loop out. That's also why we designed new versions afterwards, which have certain other design features, but that's a different story.
Yeah, it is. So anyone has any other question? Okay, actually, I have a question. It's more general, actually. Well, after watching for the first time in China what you were doing in your lab, and let me tell you, it's like not really common here in South America, especially automation in labs. So how much do you think automation and tools like biofoundries will change the everyday life of a synthetic biologist lab in the next 5 to 10 years? What do you think about it?
I mean, the concept of biofoundries is there since a while, right? There's a lot of well-organized ones. The problem is they are not necessarily making experiments cheaper. They make other types of discoveries and approaches feasible, but they need to be used and you need to have skilled people doing that. So we are on the end that I would not say we are low tech, but we try to make like something reasonable where we can operate things on a daily basis without having too much software engineering involved, where we really use the machines. Sometimes when you see the really big robots, they're just standing around and catching dust because they're too complex to operate. And also when you have those machines, they are more built for industry where you have a routine. And that means this protocol needs to run over and over again. And whenever you want to establish a new protocol, depending on the complexity, it takes weeks to months to establish that protocol with all the quality control testing. So that means in a lot of scenarios, things like this are not good for academics or research lab, because we always know that now this time I want to change this a bit and the next time this one. And I've realized there's now a completely different buffer or there's a new enzyme, and already having the changes in liquid classes means you need to adjust your pipetting steps because different liquid classes kind of are differently handled, or you have like a cooling step or a heating step, so it gets relatively complicated at certain points.
Yeah, yeah. Thank you, Daniel. Any other question in chat, or maybe someone wants to talk? Okay. Well, my last question. Sorry, Daniel. Are you currently exploring new potential applications in mind using this Vibrio, like Jumbiao's May?
So we are looking more into like expression platforms, and we're looking into a bit of like combining our regulatory tools in this organism. So it's a bit like a mixture because we have this very unique strain now, where we have a lot of things where we can go into basic biology, doing synthetic chromosomes or maybe synthetic genomes, and also maybe using this as a specific tool to be able to build really large DNAs in a faster way. Because so far we have done that with yeast, but yeast from transformation to being able to extract DNA is 2 days. With Vibrio, it's 1 afternoon. So in theory, if you make it in a good way, and if the things work, we were hoping to work, we can build large DNA much faster. But that's something where we are really in an exploratory stage. But we have a lot of different aspects we're doing with Vibrio. I also have some projects together with people in Colombia, and I'm also again in summer in Colombia for a couple of days. So we do a lot of fun things with it. It's not a main research topic. It's more like a side fun playground.
Okay. I think we have some questions in chat. Yeah. Okay. Rodrigo says, really interesting presentation. Are there opportunities for doing an internship in the lab?
Usually, yes. So we always accept people for internships. We don't have, in most of the cases, possibilities to fund that. But we always check whoever applies if there's a possibility, and we support travel grant application and so on and so forth. So this year, 2 students from Colombia made their bachelor thesis part in my lab. And in summer, there's 2 school students from Ecuador coming. So there's always someone around. And we really like the international environment, and whoever's motivated, we are always happy. Because I myself did a lot of internships when I was a student abroad. So that I think is very important for everyone.
Yeah, that's awesome. Thank you, Daniel. And I think this is the last question we have. Alan says, hello, excellent presentation. If all tRNA genes are removed from the natural genome and placed on the new chromosome, how dependent does a cell become on an artificial chromosome for its survival?
It's completely essential in the end, because you need at least 42 tRNAs. That's the 42 isoacceptors. And we have made initially the version with the 275 to then really go into the biology and see like how really isoacceptors do you really need. Can we go down to 42 tRNA genes or what? So I mean, there's a lot of biology coming in, and then you can do really unique things which were previously impossible.
Okay, thank you, Daniel. Well, actually, could you share us your email, please? So Rodrigo says, how can we contact Dr. Schindler?
Yeah, also on the website. So I think my email address is in our institute's website. But I can also put that in the chat. Give me a
Yeah,
second.
please. Okay. And Jaime says, are there opportunities for doing a PhD in the lab?
Give me a second. I'm always bad with
Yeah, sure,
focusing on
sure.
two things at the same time.
Okay, no worries.
Should be this one. There are always opportunities to do PhDs. At the moment, we have no core funding available, but there's always the chance that a grant comes in. And I usually don't advertise positions. So usually people who apply randomly or who are at the moment there when something comes in. So yeah, because the problem is if I make an advertisement, I also need to read tons of applications. While when I have like the people who write me directly to apply, then I know that they are already very motivated and they made the effort to write an application and so on and so forth. But as I said, at the moment there is no funding, but it always changes. And sometimes there are opportunities for fellowships, or there's many different options. We
Okay.
have space at the moment.
Okay, so just stay tuned. Okay, well, so that's all the time we have for questions today. Daniel, on behalf of the synthetic biology community here in Peru, we want to express our deepest gratitude. We know it's late in Germany, actually, so we truly appreciate it and the passion you share with us today. So before we let you go, we would love to take an official virtual photo to remember this wonderful session, please.